So thank you for the opportunity to present the work my lab has been doing the past year. And so with that, I will start by saying that my lab is really interested in understanding how sphingolipids are modulating diet microbiome and microbiome host interactions. And why we're so interested in this is because so the trillions of commensal bacteria that inhabit the mammalian intestine have been well associated with a variety of host physiological states and almost too many diseases to count. And so efforts now are trying to understand what are the functions of the microbiome that allow for this microbial contribution to host health. And when I think about it, or maybe when you're thinking about it, one of the most straightforward things that functions that are out there is the production of small molecules, and these small molecules that can interact with host tissue. And here I'm showing you a cartoon where small molecules produced by the gut microbiome have known functions in interacting with tissues, local tissues such as the intestine, but also having effects on tissues, far off or distal, like the brain and the skin. And so when thinking about what's allowing for this or how this is controlled, diet is a significant and unavoidable influence on the function of the gut microbiome. And we know a lot about how different macronutrient compositions of the diet can elicit different microbiome compositions and functions. However, we're still trying to understand the specifics of this, and that's needed in order for precise modulation of the microbiome using diet. And so what I like to say here is here's a handful of metabolites that are known to have really, really striking function on host metabolism. Yet there's this space for unknown. And today I'd like to fill the unknown with sphingolipids and make the case that sphingolipids are important diet microbiome host mediators. And so a lot of great work has been done to set the foundation for thinking of sphingolipids as metabolites that connect diet to microbiome composition and function. In vitro, we know that sphingolipids can promote the fitness of commensal bacteria. We also know that certain sphingolipids are toxic to some pathogens, and taking it kind of towards the human relevance and context, when you culture infant stool to understand how metabolites are interacting with the microbiome, when this is supplemented with sphingolipids, you can alter the microbiome composition based on exposure to exogenous sphingolipids. So I think it makes a pretty good case that sphingolipids could have an important role in modulating diet microbiome interactions. And then I'll talk a little bit on the other side that sphingolipids are actually produced by commensal bacteria, and sphingolipid metabolism by the microbiome is actually quite common in many host systems. Not just mammalian systems. And so what is known in mammalian systems is amazing work has been done to show the bacterially produced sphingolipids are able to modulate immune function. And additionally, we know that bacterially produced sphingolipids are also able to influence lipid metabolism in the liver. And so with that, today, I really want to focus on, I always kind of think about these connections, diet influencing microbiome and the consequences for host health. Today I'm going to focus on how sphingolipids are modulating diet microbiome interactions, but if you're ever in Ithaca or if we're ever around the table soon, hopefully, I can talk to you for hours about what the lab is doing to try to understand how sphingolipids produced by the microbiome can have effects on host health. And so I kind of hinted to it before, but we're very interested in this in the infant context, as sphingolipids can be significant components of the diets of breastfed infants. Sphingomyelin is a dominant component of the milk fat globule membrane that surrounds triglycerides and in human milk. And when you think about it this way, I mean, adults get some sort of exposure to dietary lipids, but if you do a back of the envelope calculation, infants are getting far more exposure when taking human milk. And this could have significant, and as I mentioned before, this could have significant consequences or, positive or negative effects on the microbial communities that are developing in infants across the first 6 months of life. So when I started my lab group here at Cornell, I really wanted to get into these questions involving dietary sphingolipids in the microbiome, because there's limited investigation of how human milk sphingolipids are influencing the function of the gut microbiome. And so, first you can think about what are the differences in sphingalipid composition between infant feeding modes. And so, we started looking at this to see, if there's, just a, significant implication to these two different feeding modes, which others have studied in other contexts. But we've profiled hundreds of human milk samples, and also this is our local Target or grocery store, and we just like bought all these formulas, and like, anybody will give us formulas, we'll measure the sphingolipids in them to try to get an understanding of the differences and what could be the implications for microbiome exposure. And so here I'm showing you the total ion chromatograms of human milk versus a handful of representative formulas. And us and other people have kind of shown, but I think it's really striking here that if you look at the peaks that are representative of different sphingolipid species, there are parts in this chromatogram where there's just much more complexity in human milk sphingolipids than you would find in any of your formulas. And if you actually quantify this, there are, long chain ceramides and sphingomyelins that are completely absent from, your basic formula, and also your most expensive milk fat globule membrane supplemented formula on the shelves. And so, this, implying that there might be, implications for the gut microbial metabolome based on that we can actually quantify and precisely understand if we're able to really get at the question, how are our dietary sphingolipids precisely interacting with the microbiome? And so that's the question that we set off to answer. Are they actually interacting or taken up by members of the gut microbiome? And along that line of inquiry, can we develop methods that would allow us to determine which microbes are interacting with dietary sphingolipids, and what are the consequences of these interactions? So what are the metabolites that are produced from the microbiome's interaction with dietary sphingolipids, which then would let us know, like, what is happening in this context when you have, a whole form of feeding mode where you're not getting exposure to some of these sphingolipids. And so, hypothesis is that dietary sphingolipids are being taken up by select members of the microbiome. And so there's like some sort of permissive nature of this uptake. And then microbes that are interacting with dietary sphingolipids are then actively using this to perform metabolic processes. So, so this is a picture of my lab, kind of like socially distancing around the mass spec. And I wanted to highlight that this is the work of two really talented members of my lab that I enjoy working with so much every day, Dr. Henry Lee, and who's a talented chemical biologist, and Min-Ting Lee, who is a very brave, my first graduate student in the lab, who really pioneered a lot of these techniques. that we try to come up with in order to answer some of these questions, these burning questions we had about sphingolipids and their effect on the microbiome. And so I probably don't need to explain it to this crowd, but sphingolipids are everywhere. And so when you're trying to tease out the signal of, what the exact contribution is from a particular sphingolipid, you really have to think about how to get that type of resolution from your system. And so here I'm showing you that sphingolipids are coming in from the diet, also from the host, which in this system is a mouse. Also, the microbiome is producing sphingolipids. So we really want to be able to really definitively tell which sphingolipids are coming from our particular dietary intervention. And to do this, we turn to bioorthogonal chemistry. And so we can use a modified sphingolipid that still is able to participate in the vast majority of biological processes, and this has a small functional group, an alkyne in this case, that allows us to differentiate sphingolipids that we introduce from the diet from all the rest of the ones that are hanging out there. And what we've really gotten out of the flexibility of this method is that then we can use we can use click chemistry in order to covalently link a fluorophore so that we can actually tell the location and also separate based on fluorescence and do all of the great, biological things you can do with fluorescence detection. And then also, separate from that, we can then determine products of microbial metabolism by looking at the mass difference between the native sphinganine and sphinganine alkyne. And so this gives us the tools, really, I'm showing you here, to ask who and then to ask what. And that will be more apparent as in these slides. What Minting did was she set up a system where she was orally introducing sphinganine alkyne as our candidate sphingolipid into mice for almost a week. And then isolating the microbiome content. So the cecal content is really a collection of where microbes are in the mice. And so this, you can imagine that we might think that there may be certain microbes that would take up sphinganine, and we'd be able to detect this by fluorescence microscopy. And so when looking at the confocal images of isolated microbiome contents from this mice, here I'm showing you the just general DNA stain. So this is just going to tell us in the blue channel who's there or where are the microbes. And so we have mice that were untreated and then mice that were treated or were exposed to sphinganine alkyne. And then when you look in the red channel, which is Alexaflor 647, red is what we're using to detect Alexaflor 647 azide is what we're using to actually detect the alkyne tag. And so in this case, I hope you can see that in the untreated, we don't see any red, so clean system there. And then in the treated, we see that there are particular bacteria that are taking up this dietary sphinganine. And then when you do the merge, then you're able to observe that this is selective. So not all microbes are red. You have some that are blue and don't seem to interact. And the ones that red are interacting. And so this was exciting for us because this answered one of our questions, which was, are sphingolipids coming from the diet at all important for or relevant for the microbiome? And I think this kind of visually shows that they are, but we can then still take advantage of the fact that we were able to click on a fluorophore in order to separate these microbial communities from each other. So then we can now get a population of interactors, which here are, I'm denoting in blue, like in the confocal, are non-interactors, which like in the confocal image, and then interactors, which are in red. So we detect with Alexa 647. And so right here, I'm showing you the density plots of the flow cytometry analysis, where on the x-axis is the red intensity, and you can see it in the microbiome samples of untreated mice that we have, really a pure negative population with no red fluorescence. And then in the sphinganine alkyne treated mice, that you can see this shift to a more red population that we can isolate and determine, like, we were dying to know, who are these guys? Like, who are our sphingolipid interactors that we're so excited to be able to try to characterize? And so, yes, that's the question. What is the identity? And to this, we can rely on some techniques in microbiome science. We can use 16S, the sequencing of the 16S rRNA gene, in order to give us the identity of microbes that are in a particular population. And here we're looking at microbes that are part of the positive sort, so that are interacting with dietary sphingolipids. And, so I mean, maybe some people are surprised, maybe you're not, but the majority in this particular system of sphinglipid interacting microbes are actually sphingolipid-producing microbes. So this, the genus Bacteroides is, classified by its ability to produce sphingolipids, and almost 100% of the population. And here I'm showing you relative abundance on this kind of x-axis, kind of flipped. And almost 100% is Bacteroides. And but if you zoom in, there is a minor fraction here of other taxonomic classifications, and you can still see that the top hit in this minor fraction is Prevotella, which is another sphingolipid-producing microbe of the intestinal tract. And but there are non-sphingolipid-introducing bacteria that are common kind of beneficial infant microbes, like lactobacillus or bifidobacterium, and some of us may be taking these as probiotics and such, that seem to be interacting with sphingolipids in this context. And so to get more resolution on kind of the importance of this, we wanted to try to understand, what is happening to dietary sphingolipids when they're acted upon by the microbiome. And to do this, Henry did extensive amount of comparative metabolomics in order to really decipher what were the products of microbial metabolism of dietary sphingolipids. And so here I'm showing you all the controls that were the comparisons. And so untreated, a vehicle control, and then native sphinganine treated with native sphinganine versus the sphinganine mice that were orally treated with sphinganine alkyne. And what I want to hope you appreciate here is that we're looking for kind of masses that are unique to this state, that would have a chemical formula that suggests that they contain this particular functional group. And so this is an example of a chromatogram of such a mass. And you can see that that this is unique to the sphinganine alkyne state. And it is like ceramide, if anybody's guessing out there for the mass. And just to show you, so this is indicative of a dihydroceramide that's produced by the microbiome in response to exposure to dietary sphinganine. So this was really also exciting for us. I guess the whole thing's exciting, but because it suggests that it's not just a passive kind of interaction or uptake of the sphingolipid, but an actual incorporated into the metabolic processes of certain members of the microbiome. So it's not as easy, I think, in this case, to understand the identity of who's doing these these transformations transformations as we could do with the sorting and the fluorescence and the 16S sequencing. But what we're able to do is in lab, we have kind of a catalog of these bacterial strains. And we're able to do the same kind of culturing, but in vitro, to determine which microbes in those communities may be responsible for the production of certain sphingolipid species. And here you can see that Bacteroides theta iota omicron, or B theta for short, which is our kind of model sphingalipid producer in lab, is able to produce dihydroceramides from our extensive suite. This is just an example of dihydroceramides from dietary sphinganine. And Bifidobacterium infantis down here is unable to produce certain dihydroceramides, but is able to produce certain, is able to actively transform dietary sphinganine. And so we're further trying to investigate kind of the implications of these transformations, and also, understand, beyond sphinganine, and what are, other related metabolites that and how they are interacting with the microbiome. And so in summary, I want to leave you with, sphingolipid-producing bacteria are the main classification of microbes that interact with dietary sphinganine. And we're still, ongoing experiments to understand what are the consequences. You could think, is this good or bad, or is there ever good or bad when we're thinking about microbiomes? But what are the consequences of these interactions is really important when we're thinking about trying to precisely modulate the microbiome using diet. And then the other thing I wanted to leave you with is dietary sphinganine is, can be transformed to other sphingolipids by the gut microbiome, and still trying to understand, what are the precise origins of these transformations. And then a little teaser is that in our infant cohorts, we can actually see these microbiome derived metabolites in some infant stool and not in other infant stool, and thinking about really the implications of, you need this dietary input in order to produce this microbially derived metabolite, and what are the implications for infant health when you don't have that, and how can we think about that when we're thinking about infant nutrition. And then the last thing I will share with you is just this workflow that we've called BOSS. So you can use BOSS in any system, and I hope that many use it in their system for their own problems, but it's talking about using bioorthogonal chemistry in order to trace nutrients into microbiome systems, then the ability to sort, then sequence, and use mass spectrometry in order to determine who and what. And and with that, I will just thank Henry and Minting for all their amazing work on this, and the rest of the lab, and you for your attention, and the organizers for inviting me today. So with that, I will take any questions.